Hello and welcome to IGCSE Sudarshan Studio. As we know, we are heading towards the board examinations for May-June series. So we are starting practicing of the past paper from 2025. Okay, so this is one of the latest paper, October-November 2025, and we are going to solve IGCSE Biology 0610 code/32 theory paper, and this is for the core student. We have already finished 3 questions in our previous topic or in our previous video, so we will continue the question from let's see, from the question number 4. So this was what we have. So before we start the past paper, I'll just read out the instructions because you should also read before the exam. They will give you 10 or 15 minutes before, so you have to go through the instructions here. So you need to answer all the questions. Use only, if you are writing in from the South Asia, please remember to do write only with a dark blue ballpoint pen. For diagram and graph, use the graph paper, use HB pencil basically for the graphs, okay? And for the diagram also, do write your name, center number, candidate number, and write the answers only in the space provided. You can use the rough page also, but do mention the question number and you're continuing the answer from the particular page. Okay, do not use any kind of fluid correction fluid or whitener. Do not write anything on the barcode, and you can use the calculator, but please do mention the units after your calculation. Show your calculation, because even calculation writing the formulas carry 1 mark. So the paper is for 80 marks, and let's move to the question number 4. So this was first question number, and this was from the classification topic, that is chapter 1. So we have completed this question. Then the second chapter was from osmosis topic, movement in and out of the cell or component, right? Osmosis, active transport and diffusion. The third one is from the reproduction chapter, I think so. No, this is related to the disease, basically from the plants here. And then we had for the agriculture topic, as you can see here, using the data. So we have completed this part. Okay, next moving to the fourth question. So you can check out under the playlist of biology, you will get all the videos related to the past paper of bio topic. Okay, biology IGCSE. Now let's go to question number 4. Okay, what does the 4 number question says? A figure 4.1 is a diagram of the female reproductive system. So this is from the reproductive topic, right? Now what are identification of this figure here is a female reproductive system. Now identify the letters here. A, what is your vagina, oviduct and egg cells. So we know this vagina is basically here the alphabet F. So you just need to mention here F alphabet. Oviduct, fine. Which one is the oviduct? This is also fallopian tubule. Okay, so D is the oviduct. Nowadays they are not calling it, but this is also fallopian tube. Okay, so D is also called as fallopian, AN, fallopian tube. Okay, the next one is egg cell. Egg cells are made at this point, E. Okay, and this is the egg cell, ovary area. Now, so we got 3 marks here, very easy core paper. Draw a line. Okay, label line. So whenever they ask you label, then you can label, and a letter X on figure 4.1 to show where fertilization occurs normally. So what you have to do, draw a line, right, and mention the letter X there. So you just need to, where is it, fertilization at oviduct. Okay, which is the oviduct? This one, D. So you just need to draw a line here and mention X. That's it. Use a pencil and draw X there. That's a oviduct here. Okay, label line and the letter X should be there. Show where the fertilization occurs. That's oviduct. Fine. State the name of the organ shown in 4.1 figure that produce a female reproductive hormone, and state one function of the hormone. So what's a hormone? And the organ is, here is ovary, which is producing the hormone. Okay, what is a hormone function here? Function of that hormone, ovary produces estrogen, progesterone. So we will write here development of secondary sexual characteristics. Okay, so basically ovary is releasing estrogen, progesterone. They did not ask the name here, only the name of the organ and the function. Fine, so we'll write ovary here, development because organ, and the function. 1 point for the organ, the other 1 mark is for the function, and that's how we get a 2 marks plus 3 plus 1, total 6 here. Okay, the next question is fill in the blanks. Even extended students do get fill in the blanks nowadays in the past paper or in the present paper that we are going to have. Okay, so let's see the fill in the blanks what we have here. Complete the sentence about the endocrine system using words from the list. Please remember, December, that is in the October November, they have mostly focused on the coordination topic. Okay, reproduction topic. So maybe for the Feb series we will not have much questions on these particular topic. So let's go with the fill in the blanks. Each word may be used once, more than once, or not at all. The endocrine system contains organ called, we know endocrine basically contains organs called as glands. These organs produce hormones. So we are done with the glands. Okay, some hormones help to maintain a constant dash environment. They maintain internal environment. Okay, so this is what they maintain. Our endocrine glands help us to maintain this internal environment. Okay, this is called as, the word to maintain the internal environment is homeostasis. Do you remember the chapter? Coordination and homeostasis topic is there. Okay, so this is homeostasis. The pancreas produce a hormone called as, which controls the sugar, insulin, that helps to control blood glucose concentration, and that's how you get your complete 5 marks. So we have used glucose word, we have used 1 2 3 4, right, and the homeostasis insulin part. So used once, more than once, or not at all. So you have to be very careful, read it properly, the fill in the blanks, and try to get a correct answer. Okay, let's go to the next question. Question number 5, and this is from the genetic parts. Okay, now pea plant produces seeds in pods. The color of a pea pod is controlled by a single gene. Do you know who is the father of the genetics and who has done this kind of experiment related to the pea? A famous scientist, and he was a father of a church, right? So just let me know in the comment box who is the father of the genetics here. The alleles for green is dominant and it is represented by capital G. So whenever you have such kind of question, underline some important terms there. Green is dominant and represented by G, whereas allele for yellow is recessive and represented by the small g here. Okay, now describe what is mean by allele here. Alleles are nothing but alternative versions. Version of what? Gene. So we write here alternative, either forms, or we write here versions of gene. That's it. Okay, next one. Figure 5.1 is a pedigree diagram for the inheritance of pod. Okay, can you give me the example of allele if they ask you? alternative form or version of a gene, but can you give me an example? Put it in the comment box if you know any example of allele. Fine. So we'll wait you. Let me write here. See you in the comment box for this answer, guys. Right? Okay, 5.1 is a pedigree diagram for the inheritance of pod color in some pea plants. So green is represented by a dark here circle and yellow is white. Now this is a pedigree diagram and we know green is actually dominant. Okay, so you can write this one can be either this format or this one. Okay, yellow, just for your example how we can do this one. Sometimes very easy to do. Now these are the possibilities that we have. Suppose this will be G, again capital G, or so just let's see what we have here. Question: the possible genotype for pea pod color are, okay, we got this one. So we have dominant means it's a green one, this is also green, and this is yellow. Determine the genotype of individual 1 and 2 in figure 5.1. so 1 and 2, okay, yeah, determine the genotype of individual 1 and 2 in the figure 5.1. Explain how you determined your answer for each individual. Now individual 1 genotype, what would be the genotype of 1? Of course it is a recessive one, and recessive is possible only when you have a recessive gene, right? So homozygous, it's a recessive. So what we will write here, it's a homozygous, okay, and it's a recessive. Explanation: it is the yellow, it is yellow pea which have a recessive gene, right? So this is what the explanation on this one. Next one, individual 2. Now we have the two options here, either the homozygous dominant or a recessive dominant. Now if I have this combination, let's see this part, okay, just for explanation, if I have a combination of this, so what should I get? I should get a dominant one and a recessive one, again a dominant and recessive, correct? The same will go with this also, capital and small g, and then we have again capital and small g. If I need to explain you in the chi square, this is how the possibility of first one will be, g and g, small g and small g, right? So this is what we have. I'm just explaining how to get your answer here. Fine, either you can do it this way or this way. Now if you see here, they are heterozygous, okay, as dominant allele is here, we can see that the pea will be in green color, dominant thing will be represented. So a capital is always, that is a controlling factor, is a dominant one here, okay. As you can see here, what is it? Yeah, the green allele is dominant. So basically a dominant is there, that means it will mask the recessive one. So we have green, green, green and green. Is this condition seen here? No, we don't see such kind of condition in this one because we have dominant and recessive. So the option that we get now is this one. So basically if I just this is what we will get, okay. Why? I'll give you the answer here. Let's put it in the chi square, and again we'll go to the homozygous recessive and a heterozygous condition. And now if I put the thing is, the capital G should always first, so there you go, Okay, multiply this with this, and again the same thing we follow with here. Now as you can see, we got 2 recessive, and here it is heterozygous condition also. Okay, so this is the correct answer that we will get. So now you got what is the second individual represents here. So individual 2 genotype is a heterozygous condition. How can you say that it's a heterozygous? There are yellow offsprings here. Correct? Even recessive is also there, no? So there are yellow offsprings. That's the reason you will get such kind of heterozygous condition. I hope you got the answer now and the complete explanation on this, why we are not getting, why we did not guess it for the both dominant and both recessive, homozygous dominant and homozygous recessive here, rather than we have the answer for homozygous recessive and heterozygous condition. Okay, I hope the answer is clear. If you have still any doubt with the chi-square, you can ask me or write up in the comment box, and I'll ready to help you out there. Now moving to the C part, 2 pea plants with a genotype heterozygous, that is capital and small g, for a pod color are breed together. Fine. Complete the Punnett square to determine the possible genotype of the offspring and calculate the expected phenotypic ratio. Now what we will write, both the parental gametes, we will just mention this thing first, capital G and small g, capital G and the small g. So what we get, now this is easy, right? Multiply this one this, then you multiply the capital G and small g. So first with this one and go with this. Again this is done. Now moving to the, this is how you do the multiplication. Capital G should represent first and the small g. Again this has to go with this one, so we get small g and small g. Okay, so we got 1 homozygous recessive, heterozygous we received 2, and then homozygous dominant 1. So the gene expected phenotype ratio of the offspring is green. How many are there? We'll see the capital letter. This is green, this is also green, this is also green, because G is dominant capital. Okay, so basically it will represent green. So how many green are there? So we have basically 3 green. Okay, phenotype is always mentioned in the form of a number, or phenotype presence of this genotype is we mentioned in the form of this factor. Okay, this is genotype. Phenotype, we can write the color, or you can write even the number. So here it is with 3 green pods, and how many are yellow? Only 1. So 3:1 ratio it is. Okay, hope this is clear now. Okay, moving to the 6th one. Figure 6.1 is a diagram of a spongy mesophyll cell. State the name of the organ that contains spongy mesophyll cells. This is related to your leaf, plants nutrition, plant transport topic. Okay, mesophyll cells are present in the leaf. There are other mesophyll cell, example palisade mesophyll cell, right? Now you can see chloroplast is very low in this, but in palisade mesophyll cell they are very high. Now state the name of the structure JKL. Again back to the cell topic. See how this chapter is related to your chapter 2, cell organelles. J is a cell wall, a plant cell, right? So cell wall is the first one. Next, K, the large 70 to 80% composition of the cell of a plant is a vacuole. Okay, L is the cytoplasm. Fine, this is a cytoplasm. Don't confuse with the animal cell. Animal cell will have nucleus in the center. This is a plant cell, so basically the 70-80% is occupied by a vacuole. Describe one function of the nucleus. Where is nucleus? The dark spot is basically a nucleus. So what is a function of a nucleus? They contain genetic material and controls the activity of the cell. Okay, so function is they contain genetic material, right? What genetic material we are talking about? Chromosome, DNA, RNA. Okay, they contain genetic material which controls the cell activities. Now draw an M on the figure 6.1 to show where mitochondria are found. So M, we have to just draw M later. Okay, so where do you draw M? M should be not in the vacuole. It should be anywhere in the cytoplasm. So basically you can just draw like this, no arrow marks, use only pencil and just write M. So again here, please remember the note, use no arrow, okay, no labeling, do not label it, okay, do not label it as a mitochondria until they ask you, and use only pencil. Fine, they have just mentioned you to label M, so that's it, we are doing it, and that's an M here. Okay, please do note here before you draw any diagram, use only HB pencil. Next one, state the function of the mitochondria. We know that they are the powerhouse of the cell, so they help in releasing of energy, also called as adenosine triphosphate. Moving to the next one, explain how the spongy mesophyll tissues is adapted for photosynthesis. We know that the spongy mesophyll cell or the spongy mesophyll tissues here have low amount of chloroplast. So what we will mention here, 3 points we do require it. So what do we know about the spongy mesophyll tissue? They have chloroplast. So chloroplast is basically for photosynthesis. So chloroplast, chlorophyll absorbs light. Okay, what kind of shape you can find with this? They are irregular also. So they have large surface area and loosely connected with each other, due to which there is an air space. If you remember the diagram under the plant transport topic, okay, so you'll know this one. This is, just imagine that these are spongy mesophyll cells. I'll just draw here. These are palisade mesophyll cells, okay, under the epidermis, and they will have lot of chloroplast like this, okay, and below this there's a air space, and the spongy mesophyll cells you will find like this, a lot of spongy mesophills loosely packed, and in between they have the air space like this. Okay, so this is some kind of diagram you will have. If I think now you got it. Okay, these are the palisade, this is palisade, and these ones are your spongy mesophyll cells. Okay, a lot of space and then few chloroplast. So now let's go to the explanation how spongy mesophyll tissue is adapted for photosynthesis. First thing, they have chloroplast, so they perform photosynthesis. Okay, second point, cells are irregular. So what happens now? As they are irregular, they have large surface area. Okay, now third one, they are loosely packed, so more space between the cell. Okay, the more space between the cell, and due to which there is a more space between the cell created for gas exchange. These 3 points are enough for this one. So chloroplast is there, or chlorophyll, to perform the photosynthesis here. Irregular shape provide the large surface area, and as they are loosely packed, enough space is there for the gas exchange or diffusion of carbon dioxide. And that's how the adaptation of spongy mesophyll cells for photosynthesis, its adaptation. Okay. Next one. The boxes on the left show the types of digestion. The box on the right contains sentence ending. So let's draw a line to make 4 correct sentences. Chemical digestion. What is chemical digestion doing? Break down food into smaller pieces. That is our physical digestion does. Okay. Next one. Break down food molecules. Who is doing breaking of the food molecule? Food molecules they are talking about. So breaking of food molecule is chemical way. Involve chemical changes. So basically chemical only. The last one, involve enzyme. Chemicals do that, right? So chemical digestion is the definition where the food molecules are broken, the chemical changes takes place, and even involving the enzymes in them. Whereas physical digestion only break down the food into smaller pieces. There is no chemical change occurring here. Fine. Next one. Ingestion occurs in the mouth. State the name of the part of the alimentary canal where egestion occurs. Where egestion means removal, right? So we call it as anus. Part of alimentary canal, name the part of, so it's a anus. Okay. Moving to the next one. Oh, so there we have human digestion topic. 7.1 figure shows the human tooth. Identify QRS. So Q is our enamel. I'll write first here. Enamel, just identification part here. The below this enamel only you have, you are correct, it's dentine. And the last area that we are able to see which is holding the enamel here is basically the gum. So just write the answer here. Enamel. Second one is dentine. The third one is gum. State the name of 2 structures found inside the pulp. What is present inside the pulp area? This one, the pulp area, you can see. What is there inside the pulp? We have neurons or nerves, as well as blood vessels. This will be there. That was a connectivity, right? This was a pulp area where we have the blood vessels as well as the nerves. Okay. Next one. Identify the type of tooth shown in 7.1 and explain why you have made that choice. Now, we know there are molar, premolar, okay, incisor and canine. How do I remember these 4 types of tooth? I just remember the acronym ICPM. IC Prime Minister. IC Prime Minister. I stands for incisor, C stands for canine, P stands for premolar, and M stands for molar. Now, the reason behind this incisor, we know incisor and canine both are pointed one for cutting. Okay, incisor is flat like this kind of thing, whereas canine is pointed one. Both are used for cutting down of the food, cutting. Okay, whereas if we see premolar and molar here, they have grooves like this, as you can see in the image. So they are helping in grinding purpose. So what we will write here is molar, okay, flat is top, So the top area is basically flat, and top area is flat and used for grinding. Okay, that's what one thing is there. You can see multiple roots also. That's one of the reason. Okay, multiple roots. 2 points are enough. I think 1 point is for identification and 1 point you get for your explanation. So flat top and used for grinding, here you can write it. Okay, let's see, do we have any other question? No. And that's it. That's the end of our core paper 0610/32. If you have liked this video, please like with your friends, with your students, with other schools also. So please like, share and do comment, and just let me know your doubts. And please remember, this is IGCSE Sudarshan Studio. A teacher is nothing without a student, so I do need your support. Please remember this. Support is very important. A few comments, how can I improve it, any your doubts, anything you can share, but please remember, do support your teacher here. Okay, they are your friends, philosopher, guide. That's all I can say. To like this channel, take a very good care of yourself and your family, and see you in the next video. Till then, take care. Bye.